All right. Well, hello, everyone. My name is Greg. I'm a postdoc in the imaging platform at the Broad, and I'm super excited to be here today to tell you about Cytominer, which is a computational ecosystem supporting reproducible and version-controlled processing of image-based profiling experiments. And so I've been working on this project for about a year and a half now, in large part in collaboration with these wonderful scientists you see below: Juan, Alan, Claire, Tim, Aaron, Francis, Mohammed, Adeni, Hillary, Naranj, Beth, Anne, and Shantanu. So let's just jump right in. So it's helpful to think about the Cytominer ecosystem from the perspective of the entire image-based profiling pipeline, starting with, of course, experimental design and sample prep. So this involves selecting the cell lines that you'd like to profile, selecting the perturbations, plate design, and generally getting your samples ready to be imaged by your fancy microscope. So you'll acquire the images themselves and then perform some sort of image analysis in which you segment cells or identify the cell boundaries and measure morphology features in each of the single cells. The output of step 2 is then funneled into step 3, which is the actual image profiling pipeline that processes these readouts and normalizes the data. In step 4, we take those process readouts and perform some sort of analysis to discover biology in the data. So we'll find our Waldo and then release the data to enable secondary analyses. And so we can also think of the pipeline from the perspective of the data itself. So the rawest form are the level 1 images, which then get created into single cell profiles, which are then even further analyzed, and we aggregate the single cell readouts to make profiles. So these are aggregated profiles, normalized profiles, and then finally consensus signatures, which represent replicate profiles that are collapsed to a single profile per perturbation. And so the Cytominer ecosystem exists in primarily these last 2 stages, although we do have some interaction with single cell profiles, but the primary input to the pipelines are the single cell profiles. And so it's helpful to think about Cytominer as an ecosystem, which consists of a series of 2 fundamentally different types of tools. So we have processing tools, which are Cytominer database, PyCytominer, and Cytominer eval. But then we also include infrastructure tools to help with pipeline execution. This is our recipe and our weld. And so I'll talk more about each of these 5 tools in the next couple of slides, but it's worth noting here that all of them exist on GitHub with open source licenses. They're primarily written in Python and are built on top of pandas. And so here is a more detailed picture of what our image-based profiling pipeline consists of from a data perspective. So here we have level 1 images. In step 2, in the image analysis steps, you segment your cells using either Cell Profiler or some other segmentation tool, and then you read out the morphology profiles from Cell Profiler or something like Deep Profiler. We have a tool called CytoMiner database, which collects and curates and processes and validates the single cell readouts from Cell Profiler to form level 2 data. And that handoff happens to PyCytominer, which is a separate tool, which contains the common functionality of the profiling pipeline, which include aggregation, annotation, normalization, feature selection, consensus profiles, and forming consensus profiles. And so all of these steps have some built-in options, and you can customize the pipeline depending on your experiment. The output of PyCytominer are these various intermediate data types, which can be used in various ways. And most recently, we've developed a tool called Cytominer eval to very quickly evaluate the quality of the resulting profiles. And so this framework is really nice because we can anticipate improvements to the pipeline, and we can kind of implement some of these improvements in this existing framework. And so this is a pipeline that you can then abstract to make it more easily executable. And so this is what our recipe consists of, essentially experiment-specific metadata that are going to be important for various steps along the pipeline. So this includes like metadata columns that are going to be important labels for what the output profiles are going to be, like what the perturbation is, what the cell line is, as well as various options for specifics for the actual options in each pi Cytominer steps. So in aggregation, what type of aggregation to form, what type of normalization, which feature selection steps, and so on. And so this is actually abstracted even further, and it's helpful to go take a step back all the way to the full pipeline again, in which we have our image data, which is the fundamental input. We apply our pipeline, and then we get numbers as our output. And so this last framework is what we're calling a weld. So we make a hard link between the code, the pipeline, and the data to ensure reproducibility and to enable the data scientist to go back and actually see what exactly the code was that was executed to create the data. And so I've just listed here a couple of resources that we use for dissemination of the data. So we typically put all of our images on IDR. We put intermediate data types like single cell profiles on Figshare or Zenodo. And this entire weld procedure, which works on the bulk profiles, exists with code being version controlled on Git and the data being version controlled via Git LFS. And so this is the framework that we use in the lab and also is going to be used by the JUMP consortium, which Anne has talked about, which as many pharma partners are going to be using this paradigm moving forward. And so another reason why we're really excited about this is the fact that time and time again, we've observed in science that human accessibility drives innovation. So we've seen this in the Human Genome Project, where now sequencing a genome is cheap and relatively easy. We also see this transformation in RNA-seq data handling and processing, where a lot of these pipelines and procedures are very mature now. We've seen this in access to software. So now that biologists have software like Cell Profiler and other software tools, they can spend time studying their favorite system rather than tinkering with code. We're living right now in a time in which this paradigm of having access to data and reproducible code for biomedical data scientists is really going to drive a lot of innovation in the future. And so with that, I'd like to thank you for your attention, and I'm really looking forward to any questions or discussions that we might have. And I just have a note here that contributions are welcome and encouraged, and there's no requirement that you actually have brown hair, just that you come motivated and with a smile and ready to learn and contribute. And thank you for your time.